Yeah, thank you so much for the kind introduction. Thank you for inviting me. I usually have a tendency to overpack my talks. This is, I think, the first time I have not done that. You're welcome. You're going to have a really long second coffee break. Any questions you have, go ahead, shout at me, ask anything you don't understand. I will say I'm going to be touching upon some quite technical stuff without actually going into the details. I am also the wrong person to ask about those details, but I will tell you who can answer your question. And if we have a lot of time, we might actually text people live and ask. All right. So I will talk about exactly one project today. This project has been in the making since 2017, when I first started collecting the EEG data and behavior, of course, for this project. It has not gone quite exactly the way I expected, but that's also how life gets interesting, right? So we're going to talk about this project today, and the title that I gave to my talk is Using Deconvolution to Control for Component Overlap Confounds in EEG Correlates of Decision Making. As a quick roadmap, so these are mountains with a path and the sun, in case you could not identify the icon. I'm assuming that you all do very different things, and that's probably not what I do, so I'm going to tell you a little bit about what I do, how I think about the stuff that I'm going to tell you, specifically where I think the field of value-based decision-making is right now, and where I came in with my project that I was doing, and how EEG has been used in the field at the starting point of this project. Then we're going to quickly run into issues with that, and I'm going to explain a little bit what the problem is with patterns and averages, and I'll introduce you to component overlap if you are not familiar with it. And then I will bring you the solution to the component overlap problem. Here, that's where the kind of like technical stuff comes in. I'll give you intuitions, but that's as far as I will go. I can tell you how to actually run things in MATLAB, but I have not written that code. Okay, and then after walking through deconvolution and why it's important and how we can do it, I'm going to give you a little bit of a take-home and tell you about some other tools that I think are really, really helpful and that we should be using moving forward. Okay? Brilliant. So that's what we're up to. All right, now to my talk. I study value-based decision-making, and when we think about value-based decision-making, we typically think about situations like that. A person is faced with a set of options. Conveniently, in research, it's often two, because that's easily to describe. They have two options, and they have to decide which one is better. And so what we think is going on is that people first go through something that we call valuation. So they come up with some sort of value that they assign to each of those options, right, based on certain attributes that they might have or how they feel about them and stuff like that. They will then, after having assigned those values to those options, compare those options. And that will give us things like these choice curves, where people are more likely to choose the option that is better, of course. So there was a pointer here earlier. I thought it was yours, right? I can walk around. So here we're depicting the difference in the subjective value for one item versus the other item. If that difference is positive, they're more likely to choose it. If that difference is strongly negative, they're more likely to choose the other one. And the more similar they are, the more participants choose basically at chance level. Right? Easy. And we see that same kind of like difficulty effect in response time, where if the options are very similar to each other, people are slow, and when they're very different, so the choice is easier to make, they're much faster. And we think that both choices and response times are generated from the same underlying mechanism, which is basically sequential sampling. So rather than comparing the options once, people compare the options multiple times over time and basically count how many times each one's won, and then move towards one of two boundaries. So here we have one boundary for the left option, one boundary for the right option, and as soon as one of those boundaries is hit, so sufficient evidence in favor of one of the options is accumulated, people are going to make a decision. And those types of models, and there's a couple of other parameters, so this process will be faster, so they're going to move faster towards one of the two bounds when there's a stronger signal, i.e. if the difference in values between the two options is greater. That's this one, and they're going to be slower when that difference is smaller. We call that, like I said, drift rate, and the higher the drift rate, the faster and more accurate people are going to be in the decisions that they make. But they can compensate for a low signal-to-noise ratio, for example, by increasing their threshold and accumulating more evidence before committing to a decision. And then there's other processes that go into this, such as, for example, a non-decision time, where people have to see what those options even are first, and then take some time to submit a response that has nothing to do with the decision itself. It's just other unrelated processes. And we have something like a starting point bias, but that's usually only relevant if people have some advanced expectations about the decision, or, for example, prefer pressing the right button over pressing the left button, for example. Anyway, so this is one specific example, like the drift diffusion model of sequential sampling models. There are other implementations of that. The key take home here is that you can describe the process of choosing with some type of evidence accumulation model. And people have done this in the past and then have said, well, if the brain does something like that, we should see something that correlates with something that looks like that. And so this is, for example, what's been done here. You can see the black line here. I don't know how well this is visible. There's a black line here that is the the average evidence accumulation trace, and then you have on top of that the red line that is the activity in this sets of electrodes here over the centroparietal centroparietal electrodes. And that is basically the averaged EEG trace, nicely sitting on top of that. And people have said, well, that seems to suggest that in those types of value-based decisions, people actually do something like this evidence accumulation process, and this is a correlate of that. And then they've provided a little bit more evidence and have not just looked at the average, but they have compared faster trials and slower trials, right, where the faster trials should be these and the slower trials should be those, which means there should be a steeper slope when the decision is made quickly and a shallower slope when the decision takes more time. And the EEG on those different sets of trials indeed seems to follow that same pattern, with a faster ramp for fast trials and a slower ramp for slow trials. So going into the study, we were like, great, we're going to replicate this finding, and we're going to dissociate those signals from some other signals that we think are going on, because we think that people are not just making these types of decisions when they make value-based decisions. We think they also have some sort of response to the options that they're given, right? Because for the same value difference, you can sometimes have items that are all really good. You can have items that are all really bad. In theory, these decisions should be equally difficult. They should give you the same type of response, but that's not what is happening. People feel much better when they can choose among good options than when they can choose among bad options, and that should be represented in the brain when people make these types of decisions as well. So what we wanted to see is whether there are signatures and what they look like that represent this affective appraisal of the options at hand and how they differ from those other signals. And we had some reason to believe that those types of signals exist based on previous fMRI data where these two types of signals have been dissociated spatially, so there seem to be multiple processes going on here. All right. And if you're interested in how we are thinking about this, we being me and Ahmed Haishan we've written a whole review paper about this where we explain that values are funny in a way because they probably inform what we do in many, many different ways at multiple different levels. So they might, for example, influence what decision we ultimately make between the teapot and those string lights, but they may also influence how we feel, how energetic we are moving forward, etc., etc. So what we wanted to do in the present study specifically was temporally dissociate those processes. We reasoned that, well, sure, these evidence accumulation signals basically ramp up towards the response, and they should peak at the time of the response because they're triggering it in a way. But there should be this separate signal that is tied to the stimulus onset, which is this initial response to the options at offer. And that's what we were trying to do. Find a stimulus-locked appraisal signal and a response-locked choice signal. Okay. To take the conclusion ahead, we were very wrong, and we did not find that the stuff on the left looked at all like the stuff on the top left. So that's the journey we're going to, right. Sorry, left-right is difficult. This did not look like that. All right. So how do we go about this? We recorded EEG from 39 participants as they were making these choices among consumer items that we knew how much they liked them because they had previously told us for each item in isolation using these kinds of rating scales. That's very common. That's typically how we do this in the field. And then once they had made all of their decisions, we asked them a bunch of stuff. So we asked them, how anxious did you feel making this decision? How confident are you that this is the right choice? And how much did you like the items overall? Now, we had a ton of measures, and that's too many degrees of freedom, and probably a lot of those things measure very similar things. So what we did is we applied a principal component analysis to a bunch of those different measures, including the individual item ratings, information about the choice, as well as those experience ratings. And we identified, thankfully, exactly two components. One component that is kind of like indicative of people's appraisals of those options. So that was high in green here when people said they really liked the options and when they had rated both items high individually as well as on average. And then we had the second component that tracked how difficult the choice was, how confident they were in the end, and how anxious they felt, and how extreme the values were, which also kind of tracks conflict. All right, so we had one indicator that tells us something about the choice that's being made and one indicator of how much people like these options that they're engaging with. So then we went ahead and used those principal components, those component weights, as regressors in EEG mass univariate analyses. One's of the EEG, one's locked to stimulus onset and one's time locked to the response to see what it would correlate with. Yes?
So given that you've said there's plenty of time for questions, I'm going to ask, why is chosen not pointing in the opposite-ish direction as unchosen rather than the same? Are you taking, it's like they're all constrained to positive weights, so the chosen points in more or less the opposite as unchosen.
So no, they do go in the opposite direction on this dimension, right? So basically, the higher the value of the chosen option, the easier the choice is, but the higher the value of the unchosen option, the harder it is. But if both of them are high, you're still happy because you have good options. Yeah? Cool. That's exactly the differentiation between the two components. All right, so yes, and ideally, if we analyzed stimulus and response log data separately, we would be able to differentiate the stimulus logged appraisal effects and the response log choice effects. That's what we hope to do. So, I am assuming that there will be at least some people in the room who don't understand what I mean when I say we did mass univariate analyses of EEG, so I'm going to just pop in a slide and explain real quick what that means. So, how does mass univariate analysis work? Well, it has 2 steps. The first thing is basically running a ton of regressions. Everybody knows how regression works, right? Good. So you basically run a regression analysis for each participant at each time point and each electrode on epoch data. So you get your topography, and you try in your temporal epoch, and you're trying to explain how that activation varies at each electrode and each time point across trials as a function of your regressors. So what you have is your data cube. This is all the different trials. This is all the different channels that you have. This is time. So for each electrode, each time point, each trial, you have like a little thing. And then for each trial in the trial dimension, you also have the information, well, there was a trial, and then you have the information what the weight was for appraisal on that trial, and you have the information what the weight was for choice on that trial, right? And so now you can basically solve for that, and this is what we and we basically want to have what those betas are. So how this weighted explains that, yeah. So that's what we're going to do, and we're going to do this, like I said. Hold on, something happened. Yeah, okay. So what this gives us is beta and T maps at each time point and for each regressor and participant. Okay? Okay. And you can think of generating this as similar as doing a within subject condition-wise average if you do classical ERP analyses. Intuition clear? Cool. All right, and like I said, we did this twice, once time locking the EEG to the stimulus and once time locking the EEG to the response. And once we have that, yes? This is just raw EEG. Well, it's not quite raw because we did some cleaning and filtering and that stuff before, but other than that, yes. Yeah. Yeah. Okay. Yeah, and so maybe I should say, like, in preparation of that, so you can't just throw things out, right? So if you do the ERP, classical ERP approach, what you can do is just wait, no, now I have to think. What did I do with individual artifacts? So if I do epoching to maintain this structure, oh, this has it, yes. So when I do the epoching to get the stimulus locked and the response locked things, in order to maintain the structure so that I don't lose any trials, I can't throw anything away, right? So I still do a classical like artifact detection, and I'm telling EEG Lab, hey, tell me which trials have artifacts that we haven't been able to filter out or do ocular correction on properly and like whatnot. And then I get that and I just NaN out all of these trials. So then where the data would be, there's now just NaNs, and then the regression basically ignores those. Yeah? But that's important because otherwise the EEG data is no longer going to match the trial structure. Yes? Yes. I mean, there is some overlap because we include information about the decisions that were made. Right, and those decisions were made in the time where we were also recording EEG, but the PCA is on things that preceded the EEG recording and that happened after the EEG recording. this information is, yes, derived from the PCA. This is all the information that we have about the decision, how the decision unfolded, condensed. Oh, so we're not generating, oh, this is important. This is not, what we get from the PCA is not, this is the decision that is being made. It's, what we're getting from the PCA is just an index of essentially the choice difficulty.
Yeah, I get that, but you're actually reversing the EEG data from an actual trial where a subject made a decision, right?
Yeah.
And can that response disagree with what they predict in general? Right? Like, so.
Just general, like the residuals? Are you talking about the residuals, or?
Sorry, maybe I misunderstood. So this is saying, for example, that maybe in general I like apples a lot, right, from the PCA. But then somebody has a particular EEG choice where they can choose between an apple and a tomato, and actually, even though they don't like tomatoes in that particular trial, they picked a tomato.
Oh, so. Yeah, so then I should go back for a hot second. So what we're doing here is we're integrating the measures that we have on all of these. So we do have the individual item ratings. So on a given trial, we will have the value of the chosen and the unchosen item, the average value of the two, how much they said afterwards, how much they liked the items together as a whole, as well as what the difference was between the items and value, how confident they said afterwards they were in their decision, how extreme the choice value was compared to all the other choices that they made, and how anxious they said they would feel. And we have disaggregated that, or we have done a dimensionality reduction on that to basically give us two indicators. One, how good are the options on this trial, and two, how difficult is it to choose among them. So that's what we have. And we use this as regressors to see whether we can find these correlations that we would expect with EEG data over time. So, okay. All right. So we've gone through that, and then the next step after we've gotten these cubes, so we have now three new cubes for each stimulus locked and response locked data. We have the intercept cube. That's just how, on average, all the trials, what they look like. That's just the average ERP response, basically, per subject. And then we have the deviations from that for each condition. That's what our beta weights are. And we have one for every subject. That's like having one average and two difference curves for every subject. All right. But now we want to know, is this significant across subjects? So the next thing we do is we basically run t-tests in each of those columns. So across subjects, is this different from 0? And then we look at whether there's maps or clusters that we can form both in time as well as in space. And then see if any of these clusters that we can build exceed what we would get under a random distribution. If we like flipped a random number of signs, whatever, 10,000 times, and then looked at the distribution when we do that and compare these, we look at how many clusters do we get, what do they look like when that are unlikely under this distribution. Okay, so, and the group betas that we get is what you can compare to intuitively to condition-wise grant means. So that's just the average across all subjects. If you did a grant mean by condition, that's what you would get there. Okay. So Now I've told you a bunch of stuff and you forgot what we're here for, so here's a quick reminder. What we expected to see is this, right? One stimulus-locked thing that correlates with appraisal and one response-locked thing that correlates with choice. So when we look at the results, we find basically exactly that. We find, so this is stimulus-locked on the left, response-locked on the right, and then on top we have all these like grand mean beta things for appraisal, and at the bottom we have all the grand mean beta things for choice. And what you can see is that we have a choice-related appraisal effect and a response-locked, a stimulus-locked appraisal effect and a response-locked choice effect. And that's exactly what we thought we would find. Now we can look at what that looks like, the projection for that in a more familiar fashion. So this is our projected ERP. And what we see here is that the significant cluster that we found here has a central parietal distribution and it peaks at around 700 milliseconds. Okay, so I thought that was nice because that looks exactly like these emotional salience effects that have been found previously in work where they like nicely matched the stimuli for all kinds of things and just told people like random biographical information about faces, for example, about people whose faces were shown, and then when there was emotional information associated with those faces, then people would show greater LPP. And I was like, well, nice. So we've dissociated an LPP from the thing that generates the choice. Fantastic. Super nice. And then we looked at the response data. And this had, again, also central parietal component as well as a frontocentral component. And again, I thought this is nice. That is totally consistent with what people have shown in the time frequency domain. Polania and colleagues had demonstrated not only that there are these types of activity for value-based choice specifically with a more frontal and a more posterior kind of peak, but they had actually done follow-up studies where they specifically disrupted the frontal-parietal coupling, and then people suddenly made terrible decisions. So I was like, okay, that seems to be, relevant for decision making. But then when you look closer, this thing looks kind of funny, and it does not really look like what we expect. So let me unpack why that is. So first of all, you see that the lighter lines are towards the top, so greater activity for lighter lines and lower activity for darker lines. Well, that is the opposite of what we predicted. So that's kind of funny, but there's more. So this is the same plot that I showed you on the right side of my first slide, just color-coded differently because I like this better. And what you're seeing here is, again, these fast RTs with the quick ramp and the slow RTs with the slow ramp. So let's unpack this a little bit. We see that back here we have greater activity for slow compared to fast trials. We see a ramp towards the response in both conditions, and we see that the thing peaks at the same time, regardless of how fast or slow participants responded in this on this set of trials. If we compare that to our data, we basically see the opposite. So this is also just a median split, and what we see here is in this window that is comparable to theirs, we see the opposite. We see faster trials show greater activity than slow trials. We also don't really see a ramp. Some of it actually ramps down. And we see, importantly, that the peaks that should be at the time of the response, they're moving back in time. So that is not at all what I expected to see. Yes, I had all of your faces. So one reason this could be is because the response times that they had in the previous studies were super constrained. Participants had to respond within 1.25 seconds, and ours had 4 seconds. And you can see that difference in the distribution, right? Like our people just had a lot more time and they took it. Okay, so I did what a good ERP researcher does, and I plotted a ERP image plot. Here I'm showing at the top fast response times, at the bottom slow response times. Stimulus onsets are shown with this black line, and the red line that I'm plotting here is the P3 peak. So just a constant peak that always happens after the stimulus onset. And if you squint, you can see that this peak kind of follows this peak, and this thing kind of follows this thing. That's what I saw. And I was like, that's creepy. Okay. And then I was like, but this is evidence accumulation. So clearly this thing is like getting in the way. And I remembered having heard about component overlap. I think Marta Kutas has written a really, really nice paper in psychophysiology in 2015, where she unpacks that issue. So let's assume we have an evidence accumulation process that looks exactly like what we see in that previous work, where we have a quicker ramp for fast trials compared to slow trials, but then we also have this P3 thing that sits on top of that. What should that look like? So when that thing happens, will also vary as a function of RT. So for fast trials, this is going to be really close to the other stuff. It's going to be close to the response. And for slow trials, it's going to be far away. But ERPs add up, right? So what we measure at the scalp is like a sum of all the underlying activities. So if we do add these up, what we get is something that looks very much like what we saw in our data. So it could be that there is evidence accumulation. I just don't see it because it's hidden under this other evoked thing. People have argued this before. And then I remembered this other thing that I knew about from my PhD lab, which is deconvolution. Unfold was co-developed by Olaf Diemingen, who was a postdoc in the lab where I did my PhD and learned how to do EEG. So I went back to that, and also that is when Bina was brought in as a collaborator. Just a brief introduction to Unfold. What is it and what does it do? I have blatantly stolen a bunch of figures from their website. Bina said that's okay. So what Unfold does is it deconvolves overlapping events. A nice illustration they have here is basically the principle I just described. So in our continuous EEG, we have a bunch of things that are happening, right? These extend in time, and to the extent that there is overlap in the processing of one thing and the occurrence of the other, that's going to, overlap, add up, and we're going to see a mixture of the underlying processes. So now what deconvolution can do is isolate those individual responses and tell us, oh yeah, so there is this one thing that happened, but then there was this other event, and on top of that, together that's what they look like. So here is the events that added up together would generate this. And the idea of Unfold is to go back from the top line to the bottom line to understand what those individual components look like. And to then be better able to compare conditions with each other. Okay, so if you want to apply that, how does it work? Well, if you've understood how mass univariate analysis works, then that's actually not that different. The only thing that you need to change is your first level analysis. So now what we're doing is not so much like standard ERP analysis, but it's a bit more like fMRI analysis. What we're going to do is we're going to model the individual events that we're aware of simultaneously, so stimuli and responses, for example, and then we're going to add parametric modulators. So we're going to ask, does this stimulus, so what does it look like when there is a stimulus onset? What does it look like when there is a response? And how does this response that we know then, or estimate, how does that vary as a function of what the weight for appraisal is on this trial and what the weight for choice is on this trial, right? So, essentially, we're going from this thing, having two separate sets of cubes, to just having one long data file, which is thankfully how they come out. We just now have to tell it in our design matrix what is happening when, right? So we have to tell it, oh, there's a stimulus here, there's a response here, and we have to tell it what type of condition they were in. So that we can estimate both what does it look like when a stimulus occurs in our continuous EEG or a response, and also how does the thing that occurs change as a function of choice difficulty or how much they like the options. Okay. This is what this looks like in MATLAB. There's a bit more code involved, all of which should be on the repository for this project. So if you want to do it, you can check that out. You can also look at the Unfold Toolbox website. They have excellent tutorials that explain how this works. One nice thing that the link is down there to one of the tutorials. One of the nice things that it does is it spits you out the design matrix. So this is one example where, that's why I showed you the MATLAB code. So we have, this is all S10, is every stimulus onset. This is all S20, that's all the response onsets. And then for each one, we have an intercept, and for each one, we have appraisal and choice modulators. And that just gives us basically what the value is that we have in our regression for that thing. So this is still thinking at a trial level, basically. But of course, we think, and you've seen this earlier in Ilke's talk, that these things unfold in time. So each of those regressors is going to be extended in time, and we're going to estimate what is happening after this thing occurred and a little bit before. What does that look like? And the way this is done is that we have basically basis sets. So we define a specific number of basis regressors, and then we estimate for each of them what is going on and multiply that with our, what's in our design matrix. All right. So here's a nicer and massier illustration of this. So this is assuming two conditions. So we have stimulus 1 and stimulus 2, which are from different conditions. And what we're trying to estimate is basically our EEG, which should be a sum of responses to all of these things. And then in our design matrix, we have the onset for the first condition, the onset for the second condition in time, and then we have another condition 1 again. And then we can basically explain this as the sum of, so these ones and zeros will correspond to what's here, right? So you have you have the first response to this thing, you have the last response to the thing that happened before, and you still have a response to this condition B event that has happened, right? So this is the fourth, this is the fifth, and then here we have the first for this other one. This is clear in terms of what's happening in time? Okay. And the idea is that the activity we should see is associated with, is basically a sum of all of these. So we have a weight for condition A at time point 1. We have a weight for condition A at time point 5. We have a weight for condition B at time point 4, and we have some error. And everything else right now is 0 because nothing else happened. Yeah? And we do that basically for every single window that we have. And that then gives us these different weights that explain what is happening in sum on the other side. Yeah? Cool. All right. So, you can use Unfold and just do mass univariate analysis if you like. That's an option. I would only do this now to compare that to what we get after deconvolution, and that's also what we're going to do now. So you have the option to just look at what it looks like in standard mass univariate analyses. So here is like a set of betas. This is stimulus onsets, just the intercept, right? So this is what it looks like when there's a stimulus. At time 0, you have this deflection, and you have a standard set of responses. This is just what it looks like for button presses. You have time 0, and this is your standard set of responses. This is from the tutorial, by the way. And now, without deconvolution, like I said, this is just standard mass univariate analysis, like I explained earlier. And now we can compare that to the results that we get from deconvolution, where we accounted for everything simultaneously rather than epoching first. And what you can see is that there's some stark differences here, right? There's this bump here, and this thing is much bigger than like that thing, because we've subtracted something out. And this is exactly at the time where the responses would be, right? And we can do the same thing if we look at our button press thing, and this is already leading us back to our study. We can do this button press thing, and we have this giant bulk of things here. If we subtract out the stimulus onsets, that thing is much, much smaller. Okay. So, so the deconvolution does exactly that. It removes this overlap. But, of course, it also allows us, just to come back, to not just get the average curve associated with an event, but also to say, how does that vary as a function of some regressor variable? So here, for example, what they have in the tutorial is they look at saccade-related potentials as a function of saccade amplitude. So you can see that, the greater the saccade amplitude, the greater the saccade-related potential. Well, this is without deconvolution, but here you see after the onset, if you control for all the other stuff that's happening, you see this modulation here. Is that clear? Okay, cool. Yes. Mm-hmm. Well, I mean, it's the same problem. I mean, it's the same problem in mass univariate analyses. It's also the same problem. Mass univariate is just a fancy averaging, right? So if you do standard ERP analysis, you have the same problem. So every participant is going to have a slightly different waveform. So just like a single trial may not be representative of the group average, an individual person is not going to be representative of the, say, sample average, right? But you would think that there are things that are similar, and those are the things that we're looking for. And that's the reason we do the second level analysis, where we do the cluster-based permutation test to test specifically for things that are consistent across participants. Yeah. Yeah. So, I mean, I should say the PCA is done across all subjects. So I'm not doing a separate PCA for every individual subject. This is across across all subjects data. I retrieve this PCA, and then I have shared conditions, right? Then, yeah. Okay, cool. Sorry, that wasn't clear. Okay, so I thought this is the solution. I'm going to apply this method, and then I'm going to clean up all these stimulus onsets, and then I'm going to get my evidence accumulation signal. And I was partially successful. I mean, I really removed a lot of overlapping activity. That is nice. Here I'm plotting the time course like I'd shown you before. Up top, you see the original data and with the lighter background, and at the bottom, you see the deconvolved data with the darker background. And what you will notice is that nothing changes in terms of conclusions. So the results were exactly the same. It didn't matter. It's not that there was something that was causing the findings that were weird. Those findings stand, even after the deconvolution. I don't know what we're looking at here. I am now doing additional research to try and understand what is going on here. I think it may have something to do with control. I cannot rule out at this point that that is another weird kind of artifact, but I'm trying to get into this, and I'm doing additional work on this now. But the bigger question this raised, the elephant in the room, was, is it possible that you could get something that looks like evidence accumulation from component overlap alone? So I ran some really simple simulations. They're so simple that they shouldn't be allowed to be called simulations. What I did is I basically just added up curves, okay? So you've already seen this thing-ish, right? You've already seen there is a response thing and there are stimulus things. When they occur, varies as a function of RT. If I sum those up, I get these mixed overlapping curves. The one thing that I did differently in this plot compared to the plot that I showed you earlier is that I assumed that the response log thing is constant. There's no evidence accumulation signal happening. The response is always a standard, just one thing, same curve for all conditions. That's just a response. And it still looks pretty much like the data that we found, right? It doesn't look like evidence accumulation at all. But then I told you that the response time distributions that we have are super, super different from the response time distributions that have been used before. So then I asked, okay, if I change the distributions of these things, so not the response thing, but like this and when that happens, does it look a little bit more like evidence accumulation? And yeah, I mean, if you squint a little bit, maybe, things move a little bit closer to here, and this is bigger here than, more in that time range, so kinda, maybe. But then I was like, well, my response time distribution is still like way off what they usually have in these types of evidence accumulation studies. So I shifted them closer to what they found in those previous studies where they showed evidence accumulation. And yeah, we can generate a pattern from those three curves, if we add them up, that kind of looks like evidence accumulation without having any signature or like any signal that is evidence accumulation in here, other than how much these curves jitter relative to each other based on RT. I was like, okay, so yes, creepily enough, we could get something that looks like evidence accumulation just from component overlap. So since this is possible, then I was wondering next, okay, so how much of evidence accumulation in the published papers is then component overlap? So we reanalyzed a couple of data sets, including one that I recorded myself here in panel B. We had the value-based decision-making study that inspired some of the work we did. This is 3 perceptual decision-making data sets, because you may be aware, if you are an aficionado, that the CPP was first identified in perceptual decision-making as an index of evidence accumulation. They've done a ton of really good work ruling out alternative explanations, but they hadn't looked at this. And so 4 data sets that all show a version of the characteristic pattern where I think it's fair to say that the people thought about this as signatures of evidence accumulation. So when we model stimuli and responses simultaneously, those signatures of evidence accumulation go away in every single case. Okay. Some people were not super convinced because, you could maybe, if you squint, still see something here. And there was also, some doubt that this is quite right. So we then did it another way that you can try and do it, which is by residualizing the EEG. So instead of modeling, so on the previous slide, what I showed you is we applied unfold, and we said model stimuli and responses, and let the responses vary as a function of whether this was a fast or a slow trial, just median split. So maybe we induced something there that led to this and made this fall out the way that it did. So now, in order to apply a more stringent test, we did something else. So this is just the same analysis, mass univariate as before, that maintains the effect and everything. And we can apply this analysis not to the raw data, but to data where we have fitted unfold before, and we have modeled just stimuli and response intercepts. So no parametric modulators, the model doesn't know, was this a fast trial, was this a slow trial, what happened here? It only knows here was a stimulus and here was a response. That's all the model knows. So we model these intercept only, and then we residualize it. We say, okay, so now everything that you can explain with just those two intercept beta thingies, take this out and give me what's left. And I will analyze that, and I will see what I find when I do that. Okay, so we analyze the clean data without stimuli and responses. And the intuition here is similar to, like, I don't know what you've been using for ocular correction, but in ocular, there's a lot of like ocular correction methods that basically model what the eye movement looks like, and then they remove what that signal looks like, and they keep the rest, and then you can analyze that happily as a clam. Okay, so that's basically the same thing. All right, so when we do that, we get flat lines. There's no more effect. I find that pretty strong evidence that you can get signatures of evidence accumulation from just overlapping stimulus and response related activity, regardless of condition. All right. Yeah. Almost all of the code, we're still cleaning this up, but almost all of the code should be in this GitHub repo. So I should say we've done different things. So the original value-based decision-making data has been, we've used Unfold for MATLAB, but there's also a Julia implementation. So a lot of the reanalysis, the Unfold modeling has been done with the Julia implementation. Simulations have been done with the Julia implementation. The residualizing thing has been done with the Julia implementation. Then I've like played with what that spit out. Yeah, but there's versions of Unfold code for both MATLAB and Julia, so whatever hits the spot for you. I think we have examples. All right. Okay, so we're almost there. What I haven't even touched on is the main thing that Unfold was built for, because Olaf has always been really, really paranoid about eye movements and how eye movements contaminate EEG signals, and that was really one main motivation to build this thing. And he's gone to really great lengths now in a couple of papers to address this issue and to look at, okay, how much of what we're seeing is actually driven by eye movements or differences in eye movements. And, you know, I think eye movements tell us something about cognition, but if we're interested in eye movements, maybe we should measure eye movements and not EEG to measure eye movements indirectly. And here's an example of a task where, and I think there's a paper out now that is led by Lisa Spiering, actually, with Olaf Dimigenes, senior author, where they show participants face stimuli, and I forget what they have to do. Maybe they don't even have to do anything. I think these varied as a function of like emotion or expression or something. But that's not even interesting here. So he's just showing across all conditions that It matters whether you deconvolve, of course, stimuli and responses, but also saccades and refixations. Bless you. Because what you can see here is there's responses to the stimuli. There's also really strong saccade responses. And no matter how you lock it, you see those things. And if you do deconvolution, you can isolate what those look like and maybe even study them. That's what they did in Lisa's paper. But at least you're not going to misattribute something that is not related to the thing you're interested in to that thing. Okay. So the good thing is Olaf was so obsessed with this that he's developed a bunch of different tools that can help us and that I'm now using more and more to analyze my data that can help us deal with those kinds of things. So the first thing is, and that's been around for a long time now, is EEG. That's a toolbox for linking eye tracking and EEG data that also does some eye tracking analyses and then basically puts all these analysis outputs into your EEG file so that you can easily use that information to do those types of deconvolution analyses, for example. That is now integrated with OptiCat, which is essentially eye tracking informed ocular correction, where he overweights saccade inputs and has the ICA done on eye movement heavy data, basically, and then selects components based on the relationship, the covariance between EEG and eye tracking, which I find great because it takes out a lot of the subjective, choice overload that you have otherwise. And then you basically have something that you can feed into Unfold, which you've learned about today, or probably already knew, to be honest. All right, so this for me is the way to go with the things that I want to do, because you know where people also make eye movements, like a lot. Value-based decision making. And I think that. So for me personally, eye movements there are not just a source of variance, but they're actually something worth studying in their own rights and understanding how they're related to how people make these decisions, what other decisions they might be related to that might drive those types of signals that I still have yet to explain. Those are the types of questions that I'm interested in now. By the way, I'm also hiring, so if you're interested in doing a postdoc or being an RA, watch out, there will be ads. Yeah, but so long. Thanks to my co-authors and the people who helped me collect the data, the people who keep providing the software tools, and the lab that I was in when I was conducting this research, which is Amitai's lab. Any more questions?